All right, in our last lecture video of the semester, we're going to continue talking about topics that are related to this evolutionary tree that I showed you in our previous lecture. And so in this video, we're going to be focused on the topics of evidence of evolution and how that evidence is used to construct phylogenies. And then we'll wrap up by talking some more about some crucial macroevolutionary patterns. But before I dive in to talk a little more about kind of evidence of evolution and how phylogenies get constructed, I thought it'd be useful to take a moment to talk about what types of things biologists do and think about in relation to these types of phylogenetic trees. So that brings me to a subdiscipline within biology that's called comparative biology. And this subfield of comparative biology is really concerned with trying to understand the phylogenetic or evolutionary history of individuals or taxa, and the mechanisms or the patterns that drive that history. And so being able to do this depends on describing different types of organisms, different species, and then developing hypotheses about the evolutionary relationships between those different species. So I've got an example here on this slide of two beetle species that have different looking antennae, but you'll notice they have very similar overall body structure. So that would be, for example, describing these two different beetle species and trying to figure out how are these beetle species related to each other over evolutionary time. And so this whole process basically is the process of constructing a phylogeny. So a phylogeny is essentially a hypothesis, a well-supported hypothesis about the evolutionary relationships among a group of organisms. So I want to take a minute to now talk about, well, how do we actually go about the mechanics of constructing a phylogeny? And to think about this, if we were together talking about this in the classroom, one thing that I would ask you is to start off by thinking about how many different types of evidence you can come up with that could be used to construct a phylogeny. So I've already showed you an example of a phylogeny for these flightless bird species. And so this might be, one sample system that you could think about to think about, okay, well, how could we figure out the evolutionary relationship across these different bird species? What types of evidence or what types of information could we use? So it's worth it to pause the video for a moment and just think about, can you think about types of evidence that you might be able to come up with? And then after you've done that, we'll carry forward and go through some of those types of evidence. So hopefully you've had a minute to think about types of evidence that you could use to construct one of these phylogenies. And so now we'll talk more specifically about the various categories of evidence that biologists typically use. And keeping in mind that the overall idea is you want to construct a phylogeny that incorporates as many different kinds of evidence as possible so that your phylogeny is as robust or as strong as possible, as well defined as possible. So the types of information that biologists use can range considerably, really depending on the group of organisms. But one of the biggest things that biologists look for when constructing a phylogeny is things that are called homologous characteristics or homologous traits. So look for homology. So homology refers to something that is similar across groups of organisms. We've already encountered the word actually when talking about chromosomes, right? So when we talk about homologous chromosomes, those are chromosomes that are very similar to each other. They're not identical, but they have some shared connection. And there are three different major types of homologies that biologists will use when constructing a phylogeny. One type of homologous trait that biologists will use are anatomical homologies. So I have that illustrated on the slide here. Basically, that involves looking across different anatomical structures to see if it's possible to identify similar, closely related anatomical structures. So in this slide, we've got the four limbs of four different types of organisms: a human, a dog, a bird, and a whale. And if we look across all of those limbs, what we observe is we see generally the same overall number of bones that are arranged in the same general position. So two sets of bones in the forearm, and then a set of compact bones in the wrist. And we can even find those in whales as well. So that's an anatomical homology that says this bone structure must be a shared bone structure that comes from a common ancestor. But we don't stop there. Now that we can sequence different organismal genomes, we can also use genetic sequence similarities to infer evolutionary relationships. So that's referred to as examining a molecular homology. And to give you one example of how this is done, one sequence that is very frequently used across organisms is the sequence that's found within mitochondrial DNA. So the majority of, all eukaryotes have mitochondria, and so we know that structure's there, and mitochondria contains its own DNA sequence, and mitochondria are typically inherited from an organism's mother, and they're passed on from generation to generation, and so it's possible to look at the sequence of mitochondrial DNA, see how similar or different it is from the sequences in other organisms, and then you can classify organisms based on those similarities or differences. It's also possible to use some developmental information when constructing a phylogeny. So what I'm showing here on this slide, in this slide, is genes and patterns of gene expression during early development across a wide range of different organisms. So I talked a bit about some of these developmental genes when I was talking about gene regulation, and it turns out that copies of these genes can be found across a very wide range of organisms. And so what this picture is showing you here is that range of organisms, which gene sequences are present, and where those sequences get expressed over the course of development. And so it's possible to compare those not just those gene sequences, but also where and when they get expressed to help come up with inferences about evolutionary relationships between different groups of organisms. So homologous traits play a very central role in the construction of a phylogeny, but that's not the only type of information that gets used. In addition to that, biologists will also rely on information from the fossil record. So just for fun, here's a really awesome amber fossil specimen of a type of ant that's called a hell ant. It has these really wild mandibles that are totally different from the mandibles that you'd see in any living ants. And so if we didn't have these pieces of amber that contain these ant specimens, we really wouldn't know anything about this ant specimen. But since we do have it, we can use information from this specimen and incorporate that into our thinking about the evolution of other groups of ant species as well. And just to give you a second example of kind of how fossils modify the phylogenies that we construct, here's an example of a fossil that was found and described in just 2011, 9 years ago. When this fossil was found and described, what biologists and paleontologists realized was this organism is distinct from all of the living organisms that we already know about. And based on a very detailed analysis of that fossil's characteristics, what biologists figured out was this is distinct from known groups of living organisms. But it's most closely related to a particular group of living organisms. And so when that was discovered, what that wound up doing is it wound up causing us to rewrite part of what we knew about the evolutionary relationships between different groups of organisms. And specifically, because we were able to date how old this fossil was and position it in relationship to other groups of organisms, what that wound up doing was it pushed back our estimate of when this larger group of organisms diverged back by 160 million years ago. So fossil evidence plays a very important role in reconstructing these phylogenetic or evolutionary histories. And that's not the end of it. Basically, in any case, biologists strive to incorporate as many different types of information as is available to get a sense of the evolutionary relationships between and among groups of organisms. So, for example, that could also include information about where particular organisms are located, what the range of those organisms is, and so that's using information from biogeography to think about those relationships. So here's this picture of this group of North American swallowtail butterflies, and biologists absolutely factor in the ranges of these different groups when trying to think about the evolutionary relationships across these different groups. And biologists will consider other things too. They'll also consider, the behavior of those organisms as well. And you can see there's some other information included in this table about these butterflies. Those are other characteristics that can help us understand those relationships. And could be even more than that. One critical principle for construction of phylogenies is the principle of parsimony. So in a nutshell, there's agreement that the preferred evolutionary tree is the tree that requires the minimum net amount of evolution. So it's the simplest possible explanation for how we got where we are. And so here's some of the factors that go into constructing a phylogeny using what's called character-based parsimony. So in a nutshell, to create a phylogeny, you have a group of organisms, so you have taxa, and each of those groups of organisms will be described by a set of traits. And so those are referred to as characters. And then each of those characters will be in one of a finite number of states. So evolutionarily thinking, each step in constructing the phylogeny involves one change to one character state. So the goal when constructing a phylogeny is to try and find the evolutionary tree that explains the relationship of the taxa to each other with the minimal number of changes. And I'm sure this sounds really abstract right now, and I am not going to be tasking you with the project of trying to construct phylogenies, but I think it helps to kind of understand this construction process to aid in interpreting phylogenies where and when you encounter them. So I'm going to go through an example with a theoretical phylogeny. So here are a group of organisms, taxa 1 through 6, and here are 3 different traits. So stars, circles, and squares. And so for each taxonomic group, I've just noted whether they have that trait or whether they don't have that trait. And so from this information, and this is going to be kind of one of those like insert computational steps here, what you can come up with is a phylogeny that looks something like this. So this phylogeny contains all 6 of those groups, 1, 2, 3, 4, 5, 6. Note that here at the tips of this phylogenetic tree, I have an indicator about which of those characters each of these groups has. And what's been done in constructing this tree is to figure out which of those traits were possessed by a common ancestor of all of these groups, And so on the left-hand side, what you can see is there are two groups that still have that same ancestral trait, and then construction of kind of what happened past that point through evolutionary time. So the simplest number of steps to explain the evolutionary relationships between these 6 taxa involves a series of 4 different changes to these traits or to these characters. So the addition of the circle, then we get taxa 1, the addition of the square, we get taxa 2, and then a loss of the circle, that gets us to taxa 5, and then a loss of the square, or of the star, which gets us to taxa 6. Okay, so that's basically how phylogenetic trees get constructed, but it also helps to just spend some time examining some examples of phylogenetic trees to get some practice in kind of interpreting what they're telling us about evolutionary relationships between different groups of organisms. So here's a phylogenetic tree that contains a diverse group of organisms, starting out with lungfishes, and then including other things such as amphibians, mammals, lizards and snakes, crocodiles, ostriches, hawks, and other birds. So when we're reading a phylogenetic tree, what we're doing is from left to right in this image, we're kind of reading a story about what has happened through evolutionary time. And anywhere where 2 organisms are closer to each other, in other words, there are fewer branch points between those organisms, what that means is those organisms are more closely related to each other than they are to all of the other organisms in the tree. So in this particular tree, the ostriches, the hawks, and other birds are more closely related because they are all examples of birds relative to their relationship with all of these other groups in this tree. And we can kind of then read this backwards to say, okay, then nested within that, these are then next most closely related to the crocodiles. Collectively, those 3 groups are next most closely related to lizards and snakes, etc., etc. And so you can kind of see on the right-hand side of this phylogeny, this kind of gives you another sense of how these organisms are related to each other, kind of from an evolutionary standpoint. And you'll notice, so at each of these branch points, that's where something has happened with some homologous characteristic that now distinguishes this taxonomic group from the neighboring taxonomic groups. So prior to this point, all of these organisms share in common with each other that they have digit-bearing limbs, and so that's a trait that distinguishes them from lungfishes, which do not contain digit-bearing limbs. Similarly, the amphibians do not have an amniotic sac around the eggs. That's a nutrient-filled sac. So the amnion distinguishes these other groups from the amphibians, and so forth and so on. Okay, so that all sounds great, but it doesn't always go exactly how you'd like. This is another example where the reality of biology is a little more messy than the ideal is. So here's an example of 2 extremely cute organisms. So on the left is a sugar glider, on the right is a flying squirrel. Sugar glider is an Australian marsupial mammal, flying squirrel is a North American placental mammal. If we just look at these guys, what you'll notice is they share a lot of homologous characteristics, or they share a lot of characteristics in common, right? So they've got these really big ears, they've got really big eyes, they've got these flaps of skin, etc., etc. So if we were using those characteristics to infer the evolutionary relationship, we would probably say, well, these look like they're very closely related to each other. But as the names on this slide imply, and as other evidence suggests, these guys are actually not at all closely related to each other. So the abundance of evidence suggests that these two taxonomic groups actually diverged from each other a very long time ago. So their most recent common ancestor is actually something like a rat. So how do we wind up with the sugar glider and the flying squirrel looking so similar to each other? Well, what we think has happened is when you wind up with a situation where organisms have structures that look and function similarly, but all the rest of the evidence suggests that those structures have separate evolutionary origins, then what we think has happened in those cases is what's called convergent evolution. Or in other words, what's happened is these organisms have evolved in different habitats from each other, but somehow, through the process of natural selection, they've converged on very similar anatomical solutions to shared natural selection problems in their environments. So we contrast these types of structures, so similar structures but separate evolutionary structures, we contrast those with homologous traits by referring to them as analogous traits. So if you think about the word analogy, analogy refers to something that's similar, but it's not the same, right? So an analogous structure, there are superficial similarities, but those are structures that are the result of separate evolutionary processes. And it's actually kind of cool to discover situations where living organisms have independently come up with similar solutions to different types of challenges in their environment. Here's an example of another type of oddity that comes up when trying to construct phylogenetic trees. And I've got two examples of these oddities on this slide. So one is, if you look at whale skeletons, you'll notice there's a really strange feature in whale skeletons. There's this little floating bone that's not connected to anything else in the whale skeleton. And it really doesn't appear to have any kind of function whatsoever. And there's a similar thing that happens in some groups of flying organisms where they have these little dangly structures that look like maybe they were wings once upon a time, but they don't really seem all that functional for much of anything. So these are examples of structures that are referred to as vestigial structures. So vestigial structures are kind of structural remnants that don't really seem to have much of a function, but often those types of structures can provide some hints about an organism's evolutionary ancestors and that organism's evolutionary history. So for example, the appearance of pelvic bones in the whales as a vestigial structure is part of what has allowed us to infer that whales share an evolutionary history with other groups of mammals and evolved from an organism that spent time on land. We will draw a similar inference for insect groups that have these halteres instead of having full wings. That suggests that the ancestor of these insects must have been a type of insect that did have two sets of wings, not just a single set of wings. And in reality, the halteres actually do have a function, but they're definitely not wings anymore. So from an evolutionary standpoint, we'd think of those as being vestigial. Okay, so that's how phylogenies get constructed. So I want to wrap this up by talking for another moment about some of the major patterns that have been observed after constructing phylogenies while analyzing the nature of those phylogenies to kind of figure out, you know, what's happened over longer evolutionary time across different groups of organisms. So here, basically, we're looking at the product of mutation, gene flow, genetic drift, and natural selection over 3.8 billion years, and seeing what's resulted and what kinds of patterns we can pull out. There are really three major types of patterns in macroevolutionary history. So in some cases, we observe situations where there's been stasis. So cases where there haven't been major changes in the traits of organisms through evolutionary time. So I've illustrated that with a little phylogeny up here on the upper right. So this is again organized by differences in body form on the x-axis, and then evolutionary time in this case is on my y-axis. So stasis, there are some periods of time where there aren't major changes. Meanwhile, in other parts of phylogenetic trees, we do see examples of changes in characters or examples of anagenesis happening through time. So that's illustrated with this second phylogenetic tree here, where both A and B have experienced different types of anagenesis through evolutionary time. And so anagenesis can take a number of different forms. It could be fast, could be slow, could be in one direction, like with B here, or it could be back and forth. But collectively, we see different patterns of character change through time, kind of in contrast with periods where there is stasis. And then the third major type of pattern that we observe through evolutionary time is an observation of different lineages splitting into a larger number of lineages. So this is, again, that second type of speciation that can occur. And where we see evidence of a lot of lineage splitting, that process is referred to as cladogenesis. So that's the formation of clades. So here in this third graph, I've got three different clades, or three different groups of organisms. So cladogenesis, formation of those different groups of organisms. And the speed of cladogenesis can vary across different lineages. So there can be stasis and a couple of examples of cladogenesis, as shown here for taxonomic group B. Or there can be a very consistent, regular rate of cladogenesis through evolutionary time, as shown for taxonomic group A. Or there could be an alternation of stasis and then a whole bunch of cladogenesis all at once. So it's all possible. And we do also see a lot of examples of extinction through evolutionary time as well. So collectively, observations of these different types of macroevolutionary patterns have led to a theory about the process of evolution through evolutionary time. And so the overall theory to describe one type of pattern that's been observed is this theory called punctuated equilibrium. And so biologists have really been grappling with a number of these questions that don't have strong answers. One of them is, does this process of evolution just generally occur smoothly and uniformly by very steady, gradual transformations? That's actually what Darwin originally thought for the processes that he was proposing. But when we look at the fossil record, we don't always see patterns that look smooth, uniform, and gradual. So instead, it seems like something different has happened at certain points. So, for example, oftentimes it seems like once a group of species appears, then populations are stable and we don't see a lot of other evolutionary change. And so basically, what a lot of paleontologists think is that in a lot of cases there can be a burst of speciation and then a period without a lot of dramatic changes. So that collectively has then been given this name of punctuated equilibrium. So equilibrium, refers to something that's in balance, it's not changing very rapidly. Punctuated means periodic. So there's going to be periods that are in some kind of equilibrium, and then periods where there's a lot of rapid change or bursts of speciation. And this comes from a lot of observations out of the paleontological record, and we still don't entirely know why this might be happening. It's going to depend on the group of organisms, and we still don't really have a good handle on how important these kinds of dynamics are in shaping overall evolutionary history. But I put this out there just to get you thinking, that these evolutionary processes, there's still a lot that's left to be discovered about how they play out over time, and that there are different ways of thinking about these processes and how they're unfolding through time. And probably sort of the answer to this question is going to just depend on what group of organisms is under discussion. So if we wanted to go back to phylogenetic trees and take a closer look at them, here are two phylogenetic trees that provide a slightly better indication about what's going on through evolutionary time. So the tree on the left shows a situation that emphasizes punctuated equilibrium at work. So what we see here, so we've still got morphology or physiological or physical characteristics on the x-axis and then time on the y-axis. And what this is showing is going from a single morphology to, boom, two distinct morphologies, right? So no intermediates, just an immediate change to those two other morphologies. Then again, no change until again, boom, two new morphologies appear. So that's what a phylogenetic tree where there's a lot of punctuated equilibrium would look like. The contrasting situation is called phyletic gradualism. And I'm not too concerned about remembering that particular name. What I'm more concerned about is noticing distinctions between the tree here on the right compared to the tree on the left. So in cases where there's a lot of phyletic gradualism, those are characterized by changes in morphology through time, gradual changes leading to an incremental change, the start of speciation, and then gradual divergence, repeating through time. And so phylogenies that are characterized by a lot of phyletic gradualism will contain a lot of what are called transitional fossils. So that's these intermediate fossils. And it's possible to very clearly see evidence of anagenesis or evolution happening across those fossils through evolutionary time. Okay, so hopefully that's given you some tools for understanding how phylogenetic trees get constructed and how different types of evolutionary evidence is used in that construction process. So that's the process that's been used to put together this enormous evolutionary tree that is a hypothesis about the relationship among all living organisms. So you notice that started at a single origin point, and then through evolutionary time, there's been a process of radiation of formation of new, more diverse and more complex forms. And that's really where we leave off with this course. And what you'll do next in Bio 120 is you're actually going to pick up here and start to explore the biodiversity that's found across this phylogenetic tree. So you learn more about characteristics that make each of these biological groups unique and about characteristics that they share in common as well.